Welcome to the end of chapter 16. So what we're going to look at here is a technique called FISH. It's a technique for painting chromosomes in different colors in cells. And that sounds a little weird, but this is the kind of thing you can achieve. You can do this on metaphase chromosomes. So those are chromosomes which are in cells undergoing mitotic cell division. And you can create these whole chromosome paints where each of the chromosome ends up painted a different fluorescent color. And then a skilled cytologist can actually arrange those into a karyotype to look at gross chromosomal abnormalities and the like. Or you can do this on interface chromosomes where you can then map the individual chromosomes to various chromosomal territories like we looked at in our last video here. So that's what it's for. We'll look at another application or two towards the end, but let's look at how you would do this. So this technique is dependent on appreciating that double-stranded DNA is held together by weak non-covalent bonds, hydrogen bonds, and by heating the DNA up or changing the salt or changing the pH, you can get the two strands of DNA to separate. So what we're going to do is take a short piece of DNA and we're going to fluorescently label that piece of DNA. And the piece of DNA is going to be complementary to a target sequence, and that could be a target sequence in a particular piece of the chromosome that we're interested in. So let's just say we want to look at chromosome 7, or we want to look at the end of chromosome 22 and the end of chromosome 9, for example. Just keep those chromosomes in mind for now. So we take our piece of fluorescently labeled DNA, and that's called a probe. You don't need to worry about how we create the fluorescent labeling, but all we do is add a fluorescent molecule to the double-stranded DNA. So that's called the probe, and we're going to heat that up and separate it, and that's called denaturation. And that can also be done chemically too. We're then going to take our target DNA, and that could be, for example, some cells which are in mitosis. And so the chromosomes are condensed, and we can treat them chemically so that the strands of DNA in the chromosomes separate. So what we have is double-stranded probe, which has been separated into the two single strands, and target DNA, which has also been separated. So when we've kind of opened up both of the target and the probe, if we mix them together, if we add the probe to the target, wherever the target matches the probe due to the complementarity of the sequences, the probe will bind, and so that will make that piece of target DNA fluorescent as well. Then you do a bunch of washing steps, and you put it under a fluorescence microscope, like we talked about when we talked about microscopy earlier, and you can see different parts of chromosomes. So, for example, you can look to see whether one part of a chromosome has moved to another part of a chromosome. So, for example, here is chromosome 9 and here is chromosome 22. And in certain cancers, particularly in certain leukemias, chronic myelogenous leukemia, for example, part of chromosome 9 gets switched with part of chromosome 22, and that's called a reciprocal translocation. So you end up switching out this bottom piece of 9 for 22 and ending up with these structures over here. So you can look to see if that's happened using this technique FISH. So if you make a fluorescently tagged probe for chromosome 9 and for chromosome 22, and we specifically use pieces of probe DNA which match these regions in the box, we can start to see where the pieces of DNA have moved around. So if we then do the FISH for this hybrid chromosome 22, chromosome 9, which has got a piece of chromosome 22 stuck on the bottom, and then we look for chromosome 22 with a piece of chromosome 9 stuck on it, is what we can see in a cell from, say, CML, chronic myelogenous leukemia. Here's a normal chromosome 9, but here is what's called the Philadelphia chromosome. And so the Philadelphia chromosome is a chromosome which has a small part of chromosome 9. It's lost the rest of chromosome 22, so it's going to paint two colors, and you can see the two colors painting here. Here's the normal chromosome 22. So we've got a normal chromosome 9 and we've got a normal chromosome 22, but then we've had a reciprocal translocation between this chromosome here, which is called the Philadelphia chromosome now, and this chromosome 9. So chromosome 9 has got this little piece from chromosome 22. So the other green piece of this chromosome has ended up over here on this chromosome here. So we can see when these translocations have occurred in cells and use them to understand the genetic rearrangements that are going on in certain cancers. We can also look just at chromosomes in general. And this is the application of FISH to looking for trisomy 21. So here we're just painting a piece of each chromosome. Chromosome 13 here is being painted just in green, just because in this instance we know there should be 2 copies of 13 and we suspect 3 copies of 21. So you can automate this by asking what the ratio of the green spots is to the red spots. So a computer can say there are 3 red spots, 2 red spots, and 3 red spots and 2 green spots. And so this would be a case of trisomy 21, and that's one of the causes of Down syndrome. And so you can automate that, and a machine can actually read many of the FISH analyses at the same time, looking for example, trisomy 21 in cells taken from amniotic fluid, for example. So that's just a real quick introduction to the technique called FISH. That's the end of chapter 16. I'll see you when we come back next week.